From the St. Francis Yacht Club in San Francisco, this is the Wednesday Yachting Luncheon, hosted by Ron Young.
Welcome to another of our Wednesday Yachting Luncheons. How fun to have you here. It's a beautiful day in San Francisco and the bay looks beautiful. And I want to remind everybody that a month or so ago at one of our luncheons, we raised $76,375 for Daniela Morose. So congratulations and thank you everybody for being amazingly supportive. She had wanted 40 to 50 to have a full-blown campaign for the Olympics. And you might say, well, what does a person need money for when they're going to compete in the Olympics? And the answer is to be competitive. That's what she needs it for. Many other countries, specifically and especially Great Britain, have nationally funded programs. That is to say, state-supported Olympic campaigns. And so the athletes in those programs get funding from the government as opposed to in America where we citizens fund the program. And when you're going as fast as Daniela Morose, and remember, she has 6 world championships in foiling kites. And just in case you're not familiar with that, she sails about 3 feet above the water at speeds up to 50 miles an hour. And so if you want a training partner that can help you where you're out on the water, you've got to find somebody who can stay with you more than a few seconds. That means that she has to literally find the second or third best sailor in the world from different countries and then fly them into whatever venue she's practicing in so they can stay with her and train with her. She's doing exactly that. And in her most recent regatta, in 18 races, she had 11 firsts. So she's remarkably on a real remarkable improvement scale right now, and it's largely due to the great support of her yacht club, our yacht club. She is, by the way, just to be clear, and specifically, she's the most qualified Olympian we have ever sent to the Olympics, to the Sailing Olympics. And she also, to make it more amazing, she's the youngest. She just turned 23. So congratulations on that. Also, second story ripped from the headlines. We all know about the container ship that hit the bridge in Maryland, taking it down and causing a big catastrophe. And those of you who were here to listen to Joe Farrell and Mary Crowley talk about giant deep sea renovations and deep sea excavations and deep sea recoveries. He worked on the big giant wrecks of sea. Well, Joe Farrell is literally in the news all the time now because his company was picked to handle that amazing wreck on the Maryland Bridge. So that's kind of fun to have Joe picked to do that very job. So now you got to say to yourself when you look at the schedule for the Wednesday Yachting Luncheon, holy cow, we come here and we learn about yachting activities, we learn about Olympians, we learn about deep sea recovery activities, and we learn about marine biology. Well, we got to keep track of marine biology or else it's not going to be much fun out there sailing in dank bay or ocean. Our speaker today is all about that. She went to UC Davis where she got a BS in biology, biochemistry. Then she got a master's degree from UC San Diego and was at the famous Scripps Institute. Then she went to UC Santa Cruz where she got her PhD in marine biology. Then she did postdoc at the private school USC, where she got a National Science Fellowship. She's been like a rock star in the academic world all this time. And if you look at her background, you see that what she's been doing is studying the relationship between the microbiome, that is to say, all the bacteria inside of us, and us humans, because there's a symbiotic relationship between the bacteria in us. And by the way, there's more bacteria than human cells in a person. We are more bacteria than we are human. That's a way of thinking about that. And so why would you care about this? Well, because we have to adapt to climate change, and so do the fish in the ocean, and so do all marine animals, mammals, etc. So by understanding this, we might find ways that we can deal with what's happening in the climate. Regardless whether you believe in climate change or not, whatever is happening and the world is changing, we need to know how to adapt to it. So she's become an expert in that, and she's also many of us have multiple trains of our lives and careers. And just for the fun of it, she is a triathlete, and she just qualified for the World Championships in the triathlon in Nice, France. So welcome the multi-talented Nish, is the way people call her, Michele Nishiguchi. Come on up, Michele.
Because I tend to walk around a lot. So thank you, everyone in YouTube land or Zooming in or whatever, and all you that are attending the lunch. And thank you, Ron, for inviting me. I have to admit, this is the first time I've been in this club. I've walked by it. I used to live in San Francisco many years ago. I used to work at a little company called Genentech, went back when it was on its heyday, and before I went back to graduate school. And so I used to live in San Francisco over in Noe Valley. It was a great place. I miss it, actually, a lot. But today I'm going to tell you a really cool story about this interesting little squid I work on. And it's interesting because you can use it as a model to study, as Ron said, how bacteria cooperate with animals. And so there's a lot of analogies you can use with the squid and the bacteria that they have. And for us, like I said, the majority of our bodies are bacteria. And even within our bodies, if you think about all the bacteria that are in or on us, we actually have all these habitats, right? You have a microbiome that's specific for your underarms, right? And you have a microbiome that's specific for your teeth, and even separate from your teeth or your tongue, and down your GI tract, obviously, and in your feet and wherever else, And they differ quite a lot amongst those different habitats, but also even between people. And you'd be surprised, when people look at the microbiome in their guts compared to the animals, like your pets at home. You actually have a more similar gut bacterium microbiome to your dog or cat than you do to your neighbor who's about 10 blocks away that are humans, just because of you living with your animals and you're eating around them. You might be flipping the food underneath. But yeah, it's very cool. And there's been really cool studies that have done that showed that. So just think about that. Anyways, so I'm going to talk about squid. And first, I just like to acknowledge actually where I'm at University of California, Merced. I actually drove in today from the Central Valley, and so the indigenous people there are the Yachts and the Miwok. And so because I do my research there, I just like to acknowledge that I appreciate the land that they were on before the university was built. So my love of marine biology goes back to when I was a kid. My dad is from Monterey, and so his family was there. My grandma worked in the canneries there before the war. And so we always went there as a kid, and I used to pine. And my mom is not a marine person. She doesn't like water, actually. And so she's like, what is wrong with this child, Because I just want to be out there with the scuba divers, and I always want to go in the ocean. As you know, water here is very cold. I'd go out to the beach, and, your lips are turning purple, and you're like, get back in here. And you're like, no, I want to stay in the water longer, But you can look at the habitats around here. You'll recognize the ones on the lower panels because it's the kelp forest that we have. And in fact, like Monterey Bay has something like 800 species of just macroalgae alone. So really amazing habitats for lots of marine life. And I know most of you are probably on top of it, but even when you're on top of the water, and I used to teach kayaking at Monterey Bay Kayaks as a side job when I was a graduate student. It's great to talk to the clients there and say, hey, you can just pull up a big thing of bull kelp and look at all the critters that are living on or around it. And that's not even diving underneath it. So, you have all these great places. And one of the people, you guys probably remember this guy, when I was a kid, he was one of my role models. And I always say that the way I kind of have a passion for what I do, and part of it is the marine science part, you have like a role model. And every night, I think it was Friday nights, they used to have this, the Undersea world, the Undersea world, the Jacques Cousteau. And so, and I used to watch this thing I loved. I used to pretend to be diving with him in front of the TV, on the shag carpet, and so this is what I wanted to do, right? But, one of the reasons as I got older that I was more concerned about, because I had this passion for the marine environment, was what's happening. And so most of you might know this is just a cartoon diagram of kind of what we're producing a lot as humans, which is CO2. And we naturally do this when we exhale, right? We exhale CO2, and plants do it, all these other carbon sources. And in terms of decomposition, plants, when they decompose, or even human bodies, give up CO2. And a lot of it goes into the atmosphere. That's a storage place. But also a lot of it diffuses into the ocean. And at a moderate transfer is okay. But when it starts to be too much, when we're burning too many fossil fuels, and I'm not going to preach to the choir because you probably know this, it happens to have really bad effects on the environment, and particularly in the ocean. And so what we know so far, and this is data off of NASA's website, that we know that CO2 in the oceans is going up, especially if you look at, from the mid-1960s all the way up till now. And this is taken at different places just off of Hawaii. They have a very nice station out there that monitors CO2 and pH continuously. And so you can see that's been really a big, this big jump here in just in the last, 20, 30 years. And with that, you have pH increasing. So CO2, when it goes into the ocean, dissolves, it makes it more acidic. And this is not good for many marine organisms. And a lot of people, there was a really interesting story when I was driving over here on the radio about how there's going to be a pretty serious bleaching effect in the Great Barrier Reef. There was one in Florida last year, and it's looking like it's going to happen this right in the next week or so. And that's kind of an interesting effect because what happens is that you stress the animals out, the corals, and because corals are animals, and when they can't withstand temperature, they release all their symbiotic algae, and those are the things that photosynthesize and gives them sugars to eat. They can survive for a little bit, but for long-term periods of high temperature stress, they end up dying. They need the algae, and they need the bacteria that are filling with the algae too. So I'm going to get into a little bit about that later. But pH goes up, and the other thing that's bad about high pH is that most corals, they're soft corals and they're hard corals, like the brain corals. And when pH goes up, when pH goes, it gets more acidic, so it goes down, the corals can't precipitate out the calcium needed for their hard bodies. And so what happens is that they can't make the hard exoskeleton. So it's kind of like a double-edged sword where you have these two different things, temperature and pH really changing a lot. So it's kind of not so good. And then this is, again, just to emphasize the temperatures again. What's happening is that, there's been a lot of variation, and people are like, yeah, there's a lot of variation, but what's happening is those variations are slowly creeping up to go higher and higher, and the sustaining temperatures over days or even weeks is getting longer and longer. So those are the worries that we always want to think about. With that, with temperature increase, you have the ice caps on the North and South poles. And so with higher temperatures, ice is going to melt. So we notice that there's been a sea level rise. And this is some predictions of what may happen in the future over time. And again, everyone who has beachfront property, this is a long time from now, but still, it's going to be submerged property, if we don't do anything about this. And so one of the other things is that with this increase in temperature and just a couple degrees, you can see that the diversity over time, and this is a cartoon diagram, decreases just because the loss of corals. And the corals are kind of they're like the main house. Everything lives on or around them. And if many of you have probably been to places where there's coral reefs and you're diving or you're sailing above it or whatever, and you see all the fishes and all the invertebrates. And I'm one, when I dive, I'm always looking at the small things because they're really cool stuff that live in there. But that all goes away when you lose the corals. It's just a kind of a wasteland. And it's really sad to see places like that have had these major bleaching events where there's just nothing. It's just mostly like urchins and a bunch of algae. So not so good. Okay, so the thing I'm kind of getting at, and I'm not going to make this a doom and gloom talk because I don't like to do that, but I'm going to give you some possible solutions that my lab and other people that I work with are trying to kind of investigate and use. So one of the things that I mentioned about bacteria is that they're everywhere, right? They're our partners. Generally, they're good for us. So people always think of bacteria being bad, like wash your hands, and especially after COVID too, which is a virus. Wash your hands, and you need to wash your hands. But not all bacteria are bad. Like always, you send your kids out into the playground and go play in the dirt, right? And so they're really good for us. And in fact, when you have your beneficial fauna and flora in you, it prevents pathogens from coming in. And what people have seen, like when you have these really nasty pathogenic bacteria, they're better able to invade when those beneficial microbes are gone. The beneficial microbes are there to stay. They're like your partners for life. And when you get rid of them, the pathogenic ones come in and go, hey, look at all this great real estate. Let's colonize. And then they cause the sickness. So this is where we're trying to understand how those beneficial partners can help us not just be healthy, and this is all animals and plants on the planet, but also in terms of, are they good yardsticks for determining what's going to happen with climate? So they're going to be the first ones to detect what's going on. The temperature changing, is it going to stress us out? And then how can we use them to kind of tell what's going to go on in the next level? What can we do to prevent that? So this is an old comic from the New Yorker, but so I've been always fascinated with partnerships. Like symbiosis is kind of a general term. You can have things like commensal, where you're eating at the same table and you're just enjoying the meal, but you're not benefiting from it, just sitting there. You have beneficial ones where it's a shared responsibility. I always call that a marriage. It's like a lot of compatibility. There's a little give and take on each side. And the pathogenic ones are like a bad divorce, right? So it's like you get together and like, no, this is not going to work. So we're going to focus on the good ones today, the beneficial ones. And these are just some examples of some of the beneficial ones. So we have these really cool leaf cutter ants. Have you seen those in the tropics? And the reason why they're very cool is these leaf cutter ants have this great relationship with fungi. So they cut these leaves off, fresh leaves, and they take them to their dens. And you'll see in the forest, like there'll be a train of these leafhopper ants carrying these big things of cut up leaves. And they provide, the leaf flitter provides energy for the fungi. And the fungi decompose the cellulose, which the ants can't do. And then the fungi actually give off nice little chemicals that the ants can use for eating. They eat some of the fungi. And so this is like, they provide the food, and the fungi provides this beneficial kind of nutrition for the ants. So it's a partnership. Clownfish, the same way. So habitat that the corals or the anemones provide, and the clownfish kind of benefits from protection, from predators. And they also eat parasites off the anemones. So it's kind of, again, a two-way street. They also lure things in that the anemone might eat. So it's kind of a nice thing. And then corals with their algae that they use for photosynthesis. And then bees have this great partnership with flowers, right? They pollinate, and they move pollen around to other flowers, and they get some of the pollen for themselves. So all beneficial. But I'm going to talk a little bit about this one because it's a little lesser known. And I've been working with these squids for 25 years or so. And they have this really unique partnership with a bacterium, which I'm going to show you some later today. I'm always into show and tell. So they have a really cool relationship with a group of bacteria from the family. They're called Vibrionaceae, and they actually glow in the dark. And so it's pretty cool. And my lab studies everything from the ecology of the squids, like how do they get these bacteria, to what's going on at the genetic level. So I have a lot of gene jockeys in my lab right now. We're doing a lot of manipulation with moving genes around. But we're also interested in what's happening in the field, in the real world. So everything kind of, what's going on inside the animal or the organisms, both the squid and the bacteria, to what is the implications of going out in the field when they have these changes. So to tell you about the squids, there's about 35 families of squids. So squids are mollusks, good to eat, right? and two of the families have these really cool organs we call light organs because they make light. And so these are the two families here, your market squid. These are the ones that you get the calamari rings from, and they're called Lolaginidae. And then these three are representative of the bobtail squids, or dumpling squids, they call them in Australia. And they're really cute. They're kind of only about 4 centimeters in size, and they're benthic. They kind of hang out on the sand, and I'll show you some movies later on. They bury in the sand during the day. They don't make really good aquarium display animals because it's usually like buried in the sand, and you're like, where are the squid? And they're in there. And they had them when they had the cephalopod exhibit at Monterey Bay Aquarium. They had some there, and the curator for the cephalopods, he's like, I'm not going to keep them anymore because they only come out at night, and no one's in the aquarium at night. So, but they have, if you dissect the body, which is called the mantle, right here in the center, in the ventral side, you flip it over, it has this really cool ink sac, which all squids have. And inside the ink sac, with just these two groups of squids, they have, you can see the shiny part here, and that's the light organ. So the ink sac wraps around this light organ like a sandwich, and I also make the analogy of the ink sac as like a diaphragm, like on a camera. And inside that light organ are millions of these bacteria, and this is just one channel. They're like lots of little channels that go in that light organ, and a single, those little dots here, little electron-dense dots, that's a transmission electron micrograph, is the Vibrio fischeri. It's about 1 micron in size. And just to give you a kind of a scale, the N is the nucleus of the squid. That's where all the DNA of the squids, so it's, it's pretty small. And then this is about 20,000 time magnification of one of these Vibrio fischeri bacteria. It's really neat because the way the light organ is, and these bacteria inside the light organ, they use them to actually do this really cool behavior. So what happens is that the squids, and they're in the family called Symbiotic, which is why it says Symbiotic life history. Okay. We can capture them and bring them back live. So the closest ones to here that have light organs are in Hawaii. So, damn, we got to go to Hawaii to do field work. And right now we have a big project. We're doing a field work down in Southeast Australia, so we're actually getting ready to go down there in June. But you can capture them, and you can bring them back live. And in fact, the Australian species are very robust. They can survive the 36-hour transit from when we collect and put them in little like, the aquarium bags, pack them in the coolers, and then ship them back all the way to the U.S. They actually can survive. And last time we were down there, we collected about 60, and they all made it back. And then they're happy in their aquaria in the lab. But the nicest thing is that you can bring the adults back, and they will lay eggs. And many cephalopods, they only lay, like octopus lay one big clutch of eggs and they die. These guys are sequential layers, so they lay multiple clutches, and that makes us able to use them as a model in the lab. And when the babies are born, they're born aposymbiotic, so they don't have the bacteria. And they get their glow-in-the-dark bacteria from the surrounding seawater. So what we can do in the lab is we can introduce our bacteria, we can mutate the bacteria and knock out genes that make light. We can knock out genes for colonization, and then we can give it back to the squid and ask them, does that make a difference in this symbiosis? Is it better or worse? And we can make those analogies to how beneficial bacteria react in our guts or in our bodies or whatever. So it's a really great model to do that. And then the cool thing about the bacteria, the Vibrio bacteria, is not only because they glow in the dark, but also we can culture them. So many bacteria in the world, and I don't know if many of you know, in ocean water, 1 milliliter of seawater is about 1 million bacteria. 95% of those are not culturable. They're there, but you can't culture them. And so it makes it really hard to look at the genetics of them. We can kind of take the water and kind of do a metagenome where you take everybody's bacteria, but you don't really know who belongs to what, because like this whole pool of bacteria. And we can culture these and put them in the lab, and then we can do the genetic manipulation on these to ask those kinds of questions. It's a really great model to work on symbioses. So you're like, okay, so why is she so interested about these bacteria? So the squids have these bacteria. Okay, so the bacteria live in the squid because it's a great place. They get all the nutrients free. They grow about 4 times faster in the squid than they do in seawater. And the squids use it to make light. So I told you these bacteria are bioluminescent. And now, interesting light organ that they have, there's this really cool morphology. So the bacteria inside, the ink sac wraps around, right? And they can open and close that ink sac like a diaphragm on a camera. So I said these guys are buried in the sand during the day, and they come out at night. So that's what this little thing is here. They're buried, and then at nighttime they come out. So most squids are nocturnal. And you can think about squids that live in a clear tropical environment like Hawaii. Downwelling moonlight will cast a shadow below, and things that like to eat squid, which is about everything, can see them, right? They're like, oh, look, there's a nice squid there. So what these guys do, and they're really visual animals, is they match the downwelling moonlight with the light from the bacteria, and it's like the squid cloaking device. They disappear. So it's pretty cool. And if it's a new moon, that ink sac, they just open it up, so the whole light organ is exposed, and it gets rid of the silhouette. And if it's a new moon where there's no moonlight, they close that ink sac around, so there's no light coming from the bacteria. And so squid can mechanically control the light. They also do it kind of chemically as well. The bacteria are very oxygenic. That means they need oxygen to make light. So they can control the amount of oxygen that goes to the light organ. It's pretty neat, all the kind of the crosstalk. And you're thinking like, well, how does a squid know that? So we did some studies a long time ago. It was my very first PhD student. These studies where you increase light in the lab and you have a luminometer underneath the squid. And the squid tries to match it, then you drop the light down, and it goes right down, and there's a light. So you play these great tricks with your squid, up, down, and then after a couple of times, the squids get really mad at you, and then they just shut down. They're like, nope, no more, sorry, we're done for the day, no more experiments. But this is kind of a neat behavior. It's called counter-illumination. So they counter-illuminate the downwelling moonlight. So it's like, eh. But, one of the things is that I'm interested in kind of the big picture. So how does ecology, what's so important about this in the evolution of these associations? Because we can make these analogies with these squids to, what's happening in us. So I mentioned a lot about temperature, and that's a big driving force of what we're interested now in my lab is, does temperature affect if the bacteria outside, because they get these bacteria from the environment every time they're born, does that make a difference if temperature is going up? And so this is a study a while ago with one of my grad students, Will Soto, and we took 3 species of these squids. So they're kind of, one group is in the Indo-West Pacific, and the other group's in the Mediterranean. Not too bad of places to do your field work. And so we looked at Australian ones, and one from Hawaii and one from Thailand. And the ones in red are tropical. The water's relatively warm. And the one in Tasmanica it does go up. I think we found them up near Brisbane, so it's pretty warm up there, but you can find them all the way down in Hobart, which is pretty chilly down there, so they're in cold water. And we actually do a lot of different ways to collect these guys. So we scuba, we snorkel, we dip net. We usually have a good time collecting squids. But they're cryptic, so you got to go at night. And sometimes, just get like in Hawaii, we're already dip netting, so it takes a while to get a bunch. But we also pull a seine net, too, in Australia. We can get quite a few. So there's essentially, come some effort, but we can bring them back to the lab, like I said. In France, we actually can do a bunch of other things. I work out of a marine station called Bagnols-sur-Mer. It's near the Spain-France border, right, the base of the Pyrenees. Great fortified wine there. So after you have your field work, you can go have a nice glass of wine. But we dive there, and you can see in this part here, they're kind of hiding in the sand. But at the same time, they've got the equipment to measure conductivity or salinity, temperature, and depth, so we can actually get all the physical measurements at the same place we're collecting squid. That makes it nice. And one of the things we can do, and this is just kind of to show you the variation of Vibrios. So everyone thinks that bacteria everywhere in the ocean, and they're all the same. And no, they're not. So this is just to show you kind of a snapshot of what the genotypes of these different Vibrio bacteria in these squids look like. So each one of those circles represents a genotype, like with a little difference in DNA from the next circle. And the colors represent where we collected from. And so this is just actually collecting between Hawaii, Thailand, and Australia. And you can see that things are connected. So blue is Hawaii. The earthy colors are Australia, and the pink is Thailand. And what's really cool is that you see some of these bacteria that are Hawaii, then they're kind of, the genotypes are similar to some in Australia. There's a big break here. This is actually a big temperature break in Australia. And then a lot of other Australian genotypes. And what's really neat is that you have these other ones here that then goes to Thailand and back to Hawaii. So there's a lot of transport going between Hawaii to Thailand and back with just these bacteria. And a lot of that's driven by currents. So as you sailors know, currents can have a lot of effect on where you go. And so same with the bacteria, because they're kind of at bay with where the water currents are. One of the other things I should mention is our squids, what they do is they use these bacteria at night to do this counter-elimination. And then during the day, they bury in the sand. And when they bury, they don't need the light anymore. So what they do with sunrise is they vent out 95% of those bacteria out. And 5% remains in the light organ, which then recolonizes the light organ. So it's this kind of, they're pushing these bacteria in the environment every morning. And then those bacteria are viable, they're alive, and they can recolonize another juvenile squid. So what you see is that a lot of populations, even though, like I said, it's water, there's a lot of it, the bacteria tend to stay kind of close to where their squid hosts are. And they actually have these really cool adaptations that make them the one and only. So, there's not like I said, there's 10 to the 6 bacteria per milliliter of seawater. These squids find the Vibrio species that they want. And I make the analogy of you're at a football game and the beer guy is way over there and you're here and you're trying to flag him down like, hey, come over here. And that guy has to come find you. And that's how a bacterium is trying to find its host squid when it's born. It's like, what are the mechanisms to signaling? And so my lab studies a lot of that. I'm not going to go into detail of that, but there's some really cool crosstalk that goes between the bacteria and the squid. That's all happening in the field. But what happens in the lab? So one of the things we do in the lab, and this is work that's been done by one of my previous grad students, who's now assistant professor at College of William & Mary in Virginia, and a couple of my undergrads, Jennifer Guiterres and Ferdinand Rivera. And what we can do is we can evolve these things in the lab. So in the field, the squids are, pushing these bacteria out every morning, and they go and recolonize another squid. And, it'd be nice to keep going back to Tahiti every year, but sometimes the National Science Foundation says, no, we can only let you go there like a couple times. And so one thing we can do is we can do this in the lab. We can take the animals, the babies are hatched in our lab, and we can give them different bacteria and say, okay, if you continuously do this over time with, say, a bacterium that's from Hawaii, and we put it in an Australian squid, and we do this many, many, many times, what happens to that evolved mutant? It's not really a mutant, it's just that you've taken it from a different area and you're putting it to a different host. And so we've seen that this changes. So this is just to give you an idea. This is looking at growth over high salinity of the two strains that we start out with. These are identical. The red one is just marked with an antibiotic marker so we can differentiate it from. Oops, is it almost time? Oh, okay. And this is after 2,000 generations. So just starting those two strains, and this is showing you 6 different ones. The evolution over time is different. And evolution is not a single trajectory. Like every strain can be different depending on what type of evolutionary trajectory track it takes. So it's kind of neat that you start off with one single clone, and if you do this over time, they change. And maybe it's for better, like this would be a better growth rate, or maybe it's not so good. So it just depends on what happens genetically with those bacteria. And just to give you an example, like to look at it, so this is the bottom colony there. This is a test tube of Vibrio fischeri, which is our symbiote that we work with. Normally it looks like this smooth morph. But if you let it sit, I call this the lazy grad student experiment where they forget to wash their dishes. It forms this nice biofilm on top. Biofilms are great, at least in colonization. They're not so great when you're, looking at catheters. I was talking about catheters or whatever, but they form these little wrinkly colonies just sitting there, okay? And this is like only 24 hours or 48 hours. And you can see the diversity that happens when you let it sit for a week. So from that single clone, and these are all genetically the same, or at least what they started the same, they look all different. And so and we know that the wrinkly type is actually, there's a few genes that are responsible for that. So it's neat that just sitting there in the lab, you can look at evolution in real time. That's the beauty of working with bacteria, is that their generation time is so much faster. So that's kind of looking at just like sitting there in the test tube, kind of forcing evolution. But what about the biotic factors? And this is the last thing I'm going to end with, just to give you a point. So you guys are familiar with the characters on top, right? Cartoon that I grew up with. So there's always the coyote trying to get the roadrunner. I lived in New Mexico. They're not life-size, right? Roadrunners are about like this big, right? But there's always this battle between the predator and the prey. And the classic example in biology is the Canadian lynx and the Arctic hare. So there's this kind of, you get a lot of prey, and then the predators eat them, and they kind of catch up in population, and then they drop. So there's always kind of this up and down. And what I was interested in is on the microscopic level. So at least in terms of bacteria, our bacteria form these nice biofilms, which is on the bottom. This is a scanning electron micrograph. And that's really important for colonization. If they can't make good biofilm, they can't colonize the squid. But when they're outside the squid, they're subject to all these protozoan grazers like Tetrahymena, Rhynchomonas, these are things like little ciliates in the water. And so what do the bacteria do? They have to fight back, right? So it's this predator-prey thing, but on a micro level. So what we've been looking at is looking at these 3 different grazers and different strains of bacteria, and we have some controls in the blue, and then they get grazed. That's the red ones. And you're noticing that the red gets grazed down, except for that one on the far left. So nothing happens when you put grazers on there. It's like, what's going on? Everything else is getting grazed, but not these guys. And so when you look at what the grazers are doing, you're noticing that the grazers are doing pretty well when they graze these things down, but they're dropping a little bit, and especially in that first one where you don't see any effect on having a grazer there, but the grazer is actually decreasing in number. And so what we think is that certain biofilms, at least in some of our Vibrio strains, they actually secrete a toxin to kill those grazers, so they're fighting back. And you can see that this is a scanning electron micrograph. Those little paramecium like here, they're getting dissolved, and then after they die, the bacteria eat them. So it's like, hey, go bacteria. They're able to combat the grazers just by secreting them and killing them. So it's kind of a neat thing in terms of, and I have some students that are looking at this over time and trying to figure out what genes are responsible for making those toxins. And that's what this is right here. We can look at proteins, and there's one component that's a violacin-like component that we know is responsible for killing those protists. Kind of neat stuff. So this is kind of the molecular work we're doing this. So I'm just kind of giving you a snapshot of like kind of the interplay between these two partners and what we can learn from just a really cool little squid about how bacteria interact with the host, and looking at kind of trade-offs and different kind of things in terms of social behavior. They can all relate to this model. And in a bigger system, and I'll just introduce this really quickly, recently UC Merced has been awarded this $12.5 million grant through the National Science Foundation called the Biology Integration Institute. So these are big BII grants that the NSF is interested in looking at. And ours is focusing on beneficial microbes and climate change. And we're using 3 models, so I'm the lead on this grant. We're using the squid model, but we also are using these cool insects called leafhoppers, and they're actually found in Hawaii over a big gradient going on the top of Mauna Kea all the way down. And they have nutritional bacteria. And then Aptasia, which is a sea anemone, it's a real great model for working, making analogies to coral. We don't want to actually work on the corals, so we don't want to kill the corals. But the Aptasia are like little anemones that we can do the same types of experiments without damaging corals. And so inside, actually, we're not only just doing the research, we do a lot of translational science in terms of understanding these interactions between bacteria and their animal hosts. We have a lot of outreach that we do. We're going to probably do one this summer over at Cal Academy. And also trying to include a very diverse group of students who are going to be our future STEM leaders. And that's been really fun, kind of tying the science in. And I am a true believer of the payback. Like Ron was just talking about supporting people going to the Olympics or whatever, trying to support students, getting them interested in science. And so, like I said, we're using these 3 models to look at different symbiotic systems, which can lead to lots of different things. Like these days, we're not just looking at the diversity of animals, but the signaling, the molecular signaling that's going between animal and bacterium, looking at the proteins that are produced, and then looking at some modeling gene networks. And eventually what we're going to try to do is get these models to give to conservation managers to say, hey, this is what we think is happening in the field and the lab. And if you're going to put in $1 million to preserve like this area in front of the marina, what do you need to do to be able to make it successful? So the end result of Insight is we're really trying to give back to the community in terms of these land managers and marine natural reserve managers to make these really important decisions that are going to affect us for the next 20, 50, 100 years from now. So I just wanted to end with this because right now we're at a crossroads in terms of climate to make some major impacts on whether we're going to really save some of these amazing ecosystems or just let things go. And of course, a lot of it has to do with what we do in our own lives. But as scientists, we're really trying to find ways to help people understand the little things that we can do that can make a big difference. So with that, my lab's been going for about 25 years now, and I've been really fortunate to be funded by a lot of different federal agencies and also state agencies. Previously, I was at New Mexico State University for 21 years, and I've just been at UC Merced for almost 4. But also, I have a great team of students. I've had over 150 students in my lab, undergrads and grad students, and they're the ones that are the workhorses in my lab, so I just like to acknowledge them and all my collaborators from all over the great places I've worked at. So thank you very much for listening to this, and I will be happy to, I'm going to show you a quick movie and some cool bacteria, but thank you very much. I think we had the movie up. Okay, so I mentioned that they bury, right? I think this is, you have to, why don't you go ahead and do it?
So I couldn't get one of them,
Okay. Oh, that's both. Yeah.
After you show the movie, you'll sit there. I'll sit here.
Let's go with this one. So I told you they bury in the sand. So this is one of our guys. He's just throwing sand over his head, right? And they kind of they scooch down with their bodies, and then they just throw the sand over their head. And then let's see if I can bring up the other one. This one's pretty cool too. Come on. Is it going to go? Oh, how does this one go to or no? Oh, down here. Okay. Okay, so there he goes. He's scooching down the sand. This is an aquarium. You see that? There you go. And they get their 2 arms. It's like getting buried in the sand, right? He's like, you don't see me. Yes, we do. And this is why they're not good display animals, because they're in the sand during the day. And so they don't really, they don't come out during the day. Oops, it keeps going. You can see it on the, it says, stop. And then once he gets buried by the sand, he'll pull those 2 arms in and tuck them in underneath. There you go, he's covering up his eyes there. What's really cool is right at when we have the lights going on and off, so at sunset or when the lights go off, these guys, when you drop shrimp in their tank, they know. It's like the vampires coming out. They come out, dun, 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 you know. So, anyways, and then the other thing I was going to show you guys, some of these bacteria. I think, let's see. I can quite sort of that. Can you guys see that? And you can come up and look at this later too. What is it? These are bacteria streaked on a petri dish, but they're labeled with green and red fluorescent proteins. So.
Great, so people can come and look at these later on. Perfect.
Yeah, okay. Anyway, great.
Okay.
Thanks.
You're over here.
Okay, on this side.
Wonderful.
Oh, wait, this side here, right?
Thank you, Michele.
Is this on? Okay. I think
Okay, so Nish, you've given us the case that we need our microbes and they need us. Just tell us on sort of simple terms, if all the microbes in my body died, or a person's body, if you killed them all at once somehow, what would happen and on what time frame?
you would probably die within a week. Not even. Maybe.
And what would cause me to die?
So a lot of the bacteria secrete chemicals that help you either digest food or take up minerals or any kind of amino acids better. And some of them even make certain things that we can't make ourselves. So that's one way that you wouldn't get that kind of nutritional or any kind of fending off any diseases that might be there.
So they facilitate the biomechanism of us living. Is that what you're saying?
Yeah.
Okay.
Yeah.
So now help us understand the language. Tell us the differences between in the hierarchy of microbes, bacteria, and pathogens.
Okay. So the microbes is kind of a general term. So you can anything small, and bacteria would be the unicellular cells that we have, like with different groups of E. coli or Vibrio. If you kind of use the term microbe in general, you could refer to, say, a ciliate as well, but it's not really a micro. It's not a bacterium. So yeah, the difference is bacteria are single-celled organisms. Yeast are, but they got some characteristics that are more like us. So in a general term, it's kind of like everything's small.
And what's the life cycle of these little creatures?
So with bacteria, they're clonal. And so they reproduce pretty quickly. They just split, divide in two, and it's the same identical thing. With
On
Vibrios,
what time cycle?
40 minutes.
40 minutes.
Yeah, and the E. coli, it's 20.
20 minutes.
Yeah, it's pretty fast.
So they're generational. They're valuable because you can do research on them because you can quickly test your hypothesis and they regenerate quickly. Okay.
Yeah.
Give us a sense of the size of these creatures and what's the range and variation in size?
So the Vibrios, generally, most bacteria about 1 micron in size, so if you think really small in terms of, a micron, think of like in terms of the metric scale, right? So you have microns and then millimeters and centimeters, so microns really, really small, thousands.
So how many could I put on a dime?
Oh, boy. Billions.
Billions.
Yeah. And like, so when you see those colonies on the plate there, a single colony, that's probably like 10 to the 15th bacteria on one of those single dots on that petri dish. So that's a lot of bacteria. And you can think of, in your gut, and your gut's just not bacteria, I should say. Your gut has other things like ciliates and fungi. There's like a whole microcosm in your gut. Cow guts are really interesting because they have a very specific group of organisms that help them digest cellulose. So as you know, as humans and mammals, we can't digest cellulose, right? You can eat it helps you digest things, but we can't break it down. It's bacteria and fungi that help break it down. So.
Now, what's the difference or how would you describe good pathogens and bad pathogens for humans?
So,
what is it that makes them bad or good?
Oh, so a pathogen is really, that's considered bad. That term is just not a good thing for you.
So good or bad microbes?
Good microbes are ones that are going to help you, say, digest, or actually sometimes they can help speed up your metabolism. There's been a lot of work done that's actually looking at how different microbes change the way you operate in terms of your whole metabolic capacity. And with bad ones, they break down depending on where they are. So there's like with the bad E. coli. So there's regular E. coli that are fine. And then there's these pathogenic ones. You guys, I don't know if you remember the Jack in the Box ones that were like killing people when people ate from Jack in the Box. And so they were on hamburger meat. And those, what they do is they infect, they get into your intestine and they actually settle on the little cells on your gut. And they inject toxins and they cause bleeding. So that's where it's not so good anymore because then it causes a lot of stress on your system.
So how would we support good pathogens? Should we? Is there a reason why we should?
Yeah, I think that we can't live without them. I mean, they're not just on our bodies.
How do we support them?
I think for like the ones on humans, you mean? Eating healthy. Like people always say, like don't eat junk food. Like bad stuff with a lot of sugars is not very good for you. It's also not very good for the bacteria to have too much of one type of substrate. And I think having kind of a diet, say if you're just talking about gut bacteria, having a diverse diet will be much better for you than just having one type of processed type
Do
of
the
food.
little microbes eat things?
Yes.
And so if we eat something bad, that affects their diet as well? Is that what you're saying?
Yes. And it can affect your behavior. So like I said, there's been studies where whatever substrates the bacteria pick up that are either changing their rate of metabolism, it can affect your behavior and make you feel bad.
So in a general sense, you're a marine biologist. You're a specialist within the broad field of marine biology. How often do you dive?
In Merced, not too much. No, usually I'd say 2 to 3 times a year, but a lot. So like we're going to get to go in the field in a couple months, and we'll be diving almost every day. So it's really intensive when we go out in the field to look for our critters.
And how far down do you go?
So for these guys, maybe 7 to 10 meters. So anywhere between 10 to 30 feet, not too deep. They're pretty shallow.
When you go down in the water column, how deep, what's the distribution of microbes as you go down deeper and deeper and deeper? I assume there's fewer, but I don't know. Tell us.
It's pretty consistent, actually.
Really?
Yeah, and then but it's stratified. It's not all mixed. It's not like the same bacteria up on the surface. As you go down, you'll have bacteria that are more adapted to colder water. And generally, the world's oceans are about 5 Celsius and cold. But you have the surface temperatures change with season. And then, of course, as you get to the tropics, it's warmer. And so the bacteria that like warmer habitats will tend to be around there. And they won't do as well than the ones that are colder. So,
is there the same density of microbes at the bottom of the Mariana Trench than, 10 feet out of the San Francisco Bay?
I don't know that. I mean, I think that there might be, if they're cold adapted, yes, but there's less food. So this is the thing with bacteria is that they're going to follow the food source. And so where you have a lot of dead and decaying things or a lot of productivity, like a lot of phytoplankton in the water, you get a lot of bacteria there. So places that are in the dark, there's going to be less, they call it primary productivity from the phytoplankton that are making sugars from photosynthesis. And once you go below that, there's going to be a lower density of organisms in the water.
So how do they fit in the food chain in the oceans?
Bacteria?
Yes.
So they're not only decomposing dead things, but they're also supplying nutrients for other organisms like ciliates to eat. So it's kind of like they're the base of the food chain. And then it's a wraparound because they provide food sources, especially if they're in clumps, like in a biofilm. But also they decompose the dead things at the end. So they're kind of like the main food source, or the first of all the chain of events that occur, and then the very end where they decompose things.
How much bigger are krill? Give us a krill that whales eat, and these little microbes.
Oh, about 20,000 times size. Yeah, yeah, really small.
Do the krill, and who eats these microbes, or what are they? What's the principle? Who eats them? Whose diet are they on?
So there's actually some bacteria eat other bacteria, and then like the ciliates that I showed, those things, they will eat bacteria as well. So it's a little bit. It's usually the organisms that still are pretty hard to see. You need a microscope to see them, that are feeding upon bacteria, and they call those things bacteriivores. So they eat bacteria.
So what scares you in your work?
Seeing a loss of diversity. Like when I go out, so it's interesting to look at a sense of place, right? So let's say you have a young child and they go out and they see a beautiful coral reef and they go, wow, this is amazing, right? But let's say you were there as a young child 40 years ago, whatever, and you remember that reef had way more corals, way more fish. So it's a perspective in terms of time. And it's an exercise I usually give my freshman students. It's like, talk to your grandparents and go to a place where they grew up that you know well too, and ask them what that place was like, 50 years ago, 60 years ago, and how it's changed over time.
So the diminishment of biodiversity is
Yes.
what scares you.
Yeah.
If you were asked, okay, how do we save us from reducing the biodiversity? Give us 1, 2, 3.
Less CO2 emissions and preserving places that are pristine. That's kind of a hard thing to do with all the industrialization. And 3 would be to be more aware of how we treat our sewage and everything that we produce, being more aware of sustainability.
Are the marine protective areas, is that working out as a strategy for preserving parts of the ocean?
Yes, I think so. I mean, one good example around here is the Monterey Sanctuary. And so Monterey in Central California is a really amazing place. You guys are very lucky to live here because of the Monterey Bay and the diversity of all different things, algae, invertebrates, fish, whatever. And most of you probably know about the California current that comes down if you sail. And in the spring, what happens when the winds go offshore, right? All the deep water that's underneath comes up, so the water gets a little bit colder, like around now, actually, in March, April. And all that deep water has a lot of nitrogen, so the nitrogen's sinking from decomposing organisms or whatnot. And that comes up to the surface. And nitrogen's really important because it's the building block for amino acids, which is building our proteins. Everybody needs protein. So it's a heyday in the ocean around here because it's like, wow, here's all this nitrogen. We can pick it up, starting from bacteria to protists to plankton to bigger animals, and everything goes to town. It's like I always say, yeah, springtime is great for Monterey Bay having lots of sex because everything's doing it and reproducing because there's a lot of nitrogen there. So it's like we can do it now, right? So dinoflagellates are reproducing, cloning themselves. So yeah.
So are microbes counted as biomass?
Yes. Yes.
And so in the world, what percent of the biomass is oceanic and terrestrial?
So oceanic, probably about 75%. It's way more than...
What percent?
75%.
75% of the biomass of the world is oceanic.
Yeah, because there's so many organisms living, and there's so much of the ocean that covers the planet compared to the terrestrial environment.
So what did your parents do?
So this is interesting. My mom was a head ER nurse, and my dad was a grocery guy. He did not go to college. So I'm a first-gen student. My mom went to college after I was born. She went back and got her nursing degree after that. So,
what have you done that makes each of them the most proud?
wow.
If your dad was to boast, what would he say?
Following my passion. You know, so I have a funny story. Can I tell you a quick story about this? So, every kid, I think, the parents are like, you're going to, no offense to the doctors and lawyers in here, like some profession, especially if you're a first-gen kid like I was. And I was like book smart, studying biochemistry and thinking about going to medical school and working in labs. But I was like, this is not what I want to do. I mean, I like the research, but the marine stuff has always been what I wanted to do. So when I made that decision to do that instead, Both my parents are so supportive. I mean, that was the main thing. They said, no, you need to do what you want to do because you will be happy doing that. So that turned out to be very true.
That's your dad. Would your mom have the same? What would she be proud of?
I think the same thing, too. She was a little bit more stern. She still is.
Okay, so you go to bed at night, you're thinking, and you start pondering. What puzzles you?
Let's see. How bacteria have been able to communicate, not just with each other, but with animals. I mean, they can. So it's through chemical communication. How did that evolve? It's really interesting. Like how do they tell the organism that they're living in that they're doing really well, they need to be expelled, whatever. So how did that? So
how do they communicate? Mm-hmm. We know it's some method. It's a chemical signal. Is it like a pain or something like that? How does that work?
It's a chemical signal.
Uh-huh.
But how do we? It's like the language, right? Here's the language that the bacteria speak, and then here's our language. And how do we understand each other? And how did that evolve? Like how do we know that the bacteria are saying to us?
Mm-hmm. Now, this is such a fascinating person. Of course, Paul Kamen has a question, and then, well, Kathy, you go first.
I'll ask a couple, and then I'll hand the mic to Paul. So first of all, you showed that adorable video of the bacterium, going underneath the sand. They don't make really good specimens for aquaria because they're always under the sand during the day. What if you were to put artificial sand, glass, or something transparent? Would they happily burrow and then be able to be seen?
Yeah. So you could put a glass aquarium with a clear bottom, and then you could see them from underneath, for sure.
Okay,
yeah.
Good, that's good. Håkan Håkansson has a couple good questions. In what wavelength do they emit light? Blue, red, green, cyan, magenta, or other?
Great question. Thank you.
Yeah. It's a blue light, yeah.
A blue light, okay. Yeah. And he also wants to know, where is your favorite place to dive and why?
Wow. I have two. Can I say two?
Yes, you may.
So one is here in Monterey. The kelp forests are just amazing. It's like diving in a rainforest. You just have to deal with the cold. I don't like the cold. And the other place is in Raja Ampat, Indonesia. So they've got one of the most diverse areas of corals in the world. Beautiful place, yeah.
Paul Kamen, naval architect, has a question. Paul.
How much of a microscope would one need to find interesting things in a random sample of seawater taken right from the bay?
You could just get a small, we have them in our teaching labs, so 60x, the objective would be 60x, and you could see microbes in that. They'd be really tiny, like the ones we have there that I showed the pictures, that's a transmission electron microbe, a huge thing, but you could just get one of those small ones, and they're, I don't know, usually about $800-900, and our teaching labs, we house our teaching labs with those. So yeah, and you can also see big things too, obviously the bigger, so.
Going back to coral, what's the relationship between coral and barnacles? Barnacles seem to be doing really well.
Yeah. You mean on ship hulls?
Wait. I should say Mr. Bradley is a really good sailboat racer, and we do not like barnacles in the bottoms of our boats.
Right. So you think about where a barnacle settles. So, barnacles are crabs, right? Basically crabs that are upside down and then have a house around them. They're used to living high and dry. It's one of their adaptations, right? When you go in their natural habitat, in the intertidal, they're way up high. And so they can close up the shell around it, and then they can withstand desiccation. And so you look at animals that have evolved to kind of survive extreme situations like that. They're really hard to, like on hulls, they're hard to get rid of because they've just used to having a hard life. So yeah. That'll be a constant fight. I mean, I know there's been a lot of research on barnacle glue, because if we can figure out how to get rid of that or put something that the barnacles won't settle, then, obviously that would be a good thing.
John Bechtel's monitoring online. What question, John?
You were showing a diagram of some adaptation of bacteria, two different types of bacteria adapting to different circumstances or the same circumstances. Was that a picture of genetic drift or was it epigenetic adaptation?
It's genetic drift, yeah. Wow. So there is, I don't know if the crowd knows, so epigenetic is a modification, usually by methylation of the DNA, to change the way the DNA behaves. But we've done it where we've looked at the sequence of the genome and then see the genetic changes, so yeah.
So that squid that was burrowing himself, is that what that was, a squid that was going under the sand? He's burrowing himself. Who is he afraid he'll be eaten by?
Everybody.
Crabs and so on?
Yeah, mostly in Hawaii, these big lizard fish, they're like sit and wait predators with really big mouths.
Okay.
Can I tell another side story? It's really
Yeah.
funny. So we were out in Hawaii, we dip net. We just go out in like knee-deep water and collect with dip nets. And one night, we see this lizard fish with one of our squids hanging out of its mouth. And we're like, oh, maybe that was one that didn't have its bacteria. So we grabbed the fish, and it spit the squid out. And then we took the squid back to the lab. And of course, it had its bacteria in there. It's just that it was unlucky it got caught by a lizard fish, because we were thinking, well, maybe it wasn't, counter-illuminating, but it was. It just wasn't.
So how did you catch without killing the lizard fish?
Oh, we did it. We just got it because we have big nets.
Net.
So we got
okay.
the net, and then we just kind of shook it to make it spit out the squid.
How many siblings do you have?
None. I'm an only child.
The only child.
Yeah.
So tell me something funny in your work. Something that makes you laugh a little bit.
Or maybe like excited, I guess. So
Either one.
When I'm diving if you guys dive or snorkel or whatever, seeing your organism a study, so in the case of squids, in the field, like not in the lab, doing its thing, whether it's burying in the sand or checking. So cephalopods, I maybe an opportunity to go to an aquarium and see them, but they're really visual creatures and they recognize you. And so like in the lab, my technician that goes to take care of our squids, they know that person. And when they go down to feed the squid shrimp, they come out. It's like a dog, we're waiting, So seeing in the field, they're like, you could kind of swim like this, and the squids will kind of angle towards you, so they're always checking you out. And you always wonder, like, what are they thinking about? Looking at this thing with mask and, scuba gear or whatever, and we look probably really weird. And so, yeah, I always kind of like get all excited.
So I just got back from Lanai, and at Lanai, in one of the facilities I was at that Ellison has built, there are these koi fish. There are like 60 of them that would follow me around as I walked around this beautiful resort. And so the question I have is, those koi fish are looking at me because they think I'm going to give them food, and they're all kind of herding like a bunch of little small puppy dogs. Are they talking to each other as well? What do you think?
No, they're probably just following you around. At that point, no.
Okay.
They do communicate. I mean, fish are communicating with each other all the time. But at that point, they're probably just looking, hey, there's a human, and we know that they're the ones that are going to give us our fish food.
So are we going to solve this big problem about what to do about, the climate change affecting biology on the planet?
I hope so. I'm an optimist, so I always hope that it's a kind of a behavior change for humans, and hopefully a resiliency for the organisms that are in the ocean and on land too.
So how will we will reduce the harmful chemicals which we're putting into the atmosphere? Is that the way we will solve this problem?
Partly, and I think it's also a matter of. So we're so used to building new things instead of kind of the sustainability where you can take something old and kind of use it again. So reuse instead of build anew. So it's always recycle, reuse. I think that's something that's really important to get in people's minds more so than anything else. Also, being a little bit more aware, like I said, waste. We create a lot of waste, and I'm really aware of it. In a research lab, we go through a lot of plastic, which I really hate. It's just and we've tried to figure out ways to reduce the waste, but because we're working with chemicals, we can't just throw it in the trash or recycle it. It's much harder to do that. So a lot of companies are looking at ways to do that. Biology companies that sell us lab supplies. And so always thinking about that, trying to do the research to get the answers that we need, but also trying to figure out how we reduce and reuse our supplies that we have.
So many of us believe that to solve this giant global problem, we need global solutions, and your field may contribute to the evolution or development of such solutions. What about little microbes that eat bad things in our environment? What's the future, from your view, of that path to solution?
Oh, it's great. I mean, bioremediation is really a fantastic field. So people have discovered in places where there's been oil spills, so there are bacteria that can digest it and turn it into a less toxic chemical. That's been around for 10, 15, 20 years. And that's just due to, a toxic dump happens, and there might be microbes there. There's like a few that can survive. It's like survival of the fittest, right? And those bacteria then that can survive in that toxic dump, they will reproduce, and then there'll be more of them and more of them until you get a good population of bacteria that have that special capability of breaking down hydrocarbons. So finding those kinds of resources and then making them available to help us clean up waste, like an oil tanker dumping out.
So to help your research, you received, you guys were granted a $12.5 million grant for the National Science Foundation. Did you have a grant writer on staff to do that? How did you arrange and manage that process?
Myself and the team. So I'm the lead on that grant. I have 4 other PIs or co-PIs.
PI means what?
Principal investigator.
Okay.
And most of us are at UC Merced. We have 2 collaborators at Michigan State University. And then I work with the conservation manager for the Nature Conservancy. Mm-hmm. She's based in Monterey. And so we kind of had the good part of it with the research part by the scientists, but also we have a bunch of mathematicians. So there's biologists, mathematicians, bioinformatics people, and then conservation ecologists. So.
Vice Chairman Kathy Trafton has another question. Kathy.
Hogan's question is, which is more intelligent, a squid or an octopus?
Oh, well, I think octopus are. I would say, they're both in the same group, but so the thing about squid and octopus, so squid are mostly in the water column, except for our guys. There's a couple that are on the sand. And so they're mostly swimming and escaping predators. But octopus escaping, yeah, and they usually swim away. But octopus are always on the bottom, and if you're on the bottom, there's a lot more things that can eat you than up here, right? It's one of the reasons why cephalopods lost their shells, is to get away from predators and also to chase more things to eat up here in the water column. So octopus have to be pretty. That's the reason why they've evolved the camouflage and the able to make their skins bumpy or match their background. And so I would say their brains are about the same size, but some of the reasons why they've been able to do that more so.
And a follow-up, do you eat squid and octopus?
I do. I did not for a while. And it was one long day of dissecting a lot of squid, and then I couldn't do it anymore. And then about, 10, 15 years later, I was at a dinner actually in Vigo, Spain. And one of their best things is pulpo gallego. So that's the nice octopus with balsamic vinegar and paprika and olive oil. And not balsamic, just paprika and olive oil. And it's so good. So I started eating again.
Michele Nishiguchi, UC Merced, thank you so much for sharing your insights with the Wednesday Yachting Luncheon.
Thanks.
Help us save the Earth.
Thank you.
Great job.
Thank you.
Terrific.
Thank you.
Totally entertaining. Great. Help save the world.
This has been a presentation of the Wednesday Yachting Luncheon.